A very good morning to its audience. I am Syamim Nukman Sapiyan, and I would like to present the evaluation of coconut milk as a plant-based cheese using psychochemical analysis. In recent years, the shift toward plant-based diets have been driven by concerns over sustainability, food security, and advancement in food innovations, indicated by Hassan et al. 2022. One such breakthrough is coconut milk cheese, a dairy-free alternative that mimics the characteristics of traditional cheese through microbial fermentations. This process relies on Lactococcus lactis subspecies Cremaris, a bacterial strain widely used in conventional cheesemaking, to develop the texture, acidity, and flavor profile of the final product. Coconut milk, naturally rich in protein such as albumin, globulin, prolamine, and glutenin, provide a sustainable matrix for cheese production, offering structural integrity and nutritional benefits, indicated by Halim et al. 2023. As a consumer, demand for dairy-free options continues to grow, and coconut milk cheese presents as a promising solution that balance functionality, taste, and sustainability. One of the main difficulties in creating coconut milk cheese is replicating the texture, flavor, and melting properties of traditional dairy cheese, as coconut milk lacks of casein, the key protein in dairy cheese structure. Additionally, lactose intolerance is a major dietary concern, leading many individuals to seek dairy-free alternatives, indicated by Fascioni et al. 2021. The study is significant because it aims to enhance the texture of plant-based cheese through an optimized or ideal fermentation process, making it more comparable to dairy cheese. Moreover, it supports the expanding dairy alternative market and contributes to food innovation by offering a sustainable, health-conscious solutions. By addressing these challenges, coconut milk cheese can provide a high-quality option for those seeking plant-based and lactose-free alternatives. The objective of this analysis first is to produce plant-based cheese using coconut milk as a raw material. Second, to determine the ideal processing conditions for effective cheese production from coconut milk. And the last one, to investigate the psychochemical properties of coconut milk before and after cheese making. The literature review is focused on a key aspect of cheese production, particularly focusing on food chemistry, plant-based alternative, and the role of microorganisms. First, research by Paulos et al. 2023 highlights the importance of calcium in cheese production as it influences the coagulation process and overall yield. This is crucial in ensuring firm and stable cheese texture. Next, Craig et al. 2022 discuss plant-based cheese substitute, where powdered cellulose acts as an anti-caking agent, helping to create various forms of cheese alternatives, including spreads, slices, and blocks. This development supports the growing demand of dairy-free options product. Finally, Ahmad et al. 2021 emphasized the role of beneficial microorganisms like Bifidobacterium bifidum, Lactobacillus casei, and Lactococcus lactis subspecies lactis in cheese production. These cultures enhance texture, flavor, and overall quality, making them essential in both traditional and plant-based cheese formulations. So here the methodology. At the left side is the flowchart of bacterial culture and preparation for bacterial count, while the right side is flowchart of the cheese making. And here the list of analysis that being conducted. In this slide, we discuss the total number of colonies measured in CFU program. According to the study, the lactic acid bacteria (LAB) population during cheese production was initially around 9.0 log CFU per gram, or equivalent to 10 to the power of 9 CFU per gram across all samples. However, during storage, this population declined to a range of 6.5 to 7.0 log CFU per gram, or approximately 10 to the power of 6 to the 10 power of 7 CFU per gram. As noted by Camarillo et al. 2023, an initial LAB concentration of 2.9 times the power of 7 CFU per gram falls within the expected range for cheese production. This data highlights the natural decline of microbial population over time, which is a crucial factor in understanding cheese maturation and shelf life. Maintaining an optimal LAB count is essential for ensuring the desired texture, flavor, and preservation of the final product. The result of the texture from day 0 to day 3 at 37 Celsius for coconut milk cheese. In this slide, we examine the color changes in cheese sample during fermentation at different temperatures. According to research, ripening is frequently linked to the breakdown of protein and fat, leading to structural alteration that reflects more light, making the cheese appear lighter. This observation aligns with findings from Ferris et al. 2024. During fermentation, enzyme activity and pigment synthesis contribute to this yellowing effect, as explained by Dufosse et al. 2005. Our result shows that while MC undergoes more noticeable color changes due to fermentation dynamics, and CMC maintains a more stable color, making it a viable non-dairy alternative with consistent visual appeal. Additionally, previous research by Isaac Elijah et al. 2020 found that cow's milk cheese exhibits significant variance in color, ranging between 77 to 89 depending on aging and fat content, which align with our data. This finding emphasized the importance of color stability in developing plant-based cheese alternative. In this slide, we analyze the pH changes observed in our sample at different fermentation temperature. The results indicate that pH decreased more rapidly at 37 Celsius, which align with findings by Zota et al. 2017, showing that LAB enzymes are more effective at higher incubation temperatures, accelerating fermentation. A similar study by Harper et al. 2022 on fermented coconut milk found a pH drop from 6.35 to a range of 3.96 in a range with 4.98 at 37 Celsius, comparable to cream cheese. Traditional cream cheese made from 100% cow's milk has a pH range 4.4 to 4.9, indicated by Agarwal et al. 2008, which contribute to its tangy flavor and spreadable texture. Our result shows that MC samples exhibit a more significant pH drop than CMC, highlighting the role of fermentation dynamics in acid production. To determine the statistical significance, we perform Duncan's multiple range test. The means with the same letter in the tables are not significantly different, while different letters indicate a significant difference in pH between samples. This analysis helps us understand the variation in acidity and how different fermentation conditions affect the final product's quality. In this slide, we analyze the texture development during fermentation by observing the firmness of our samples at different temperatures. As Fernandez et al. 2017 states, coconut milk has a naturally firmer consistency due to its higher fat content and solid particles, which explain the initial firmness seen in CMC samples. The results show a significant increase in firmness over time, especially at 37 Celsius. This aligns with Lucy 2002, who reported that faster acidification and protein network stabilization occur due to increased metabolic activity of lactic acid bacteria at higher temperature. Additionally, the solidification of coconut oil at 37 Celsius may have further contributed to this increase in firmness. Duncan multiple range test was used to analyze statistical significance where means with the same letter are not significantly different. Our findings indicate that CMC at 37 Celsius exhibited the highest firmness, reaching 369.92 G-force, which fall within the range of traditional dairy cheese structure indicated by Hatching et al. 2024. Furthermore, a firmness measurement of 339.9 aligns with value reported by Ivanov et al. 2018 for casual cow's milk cheese, who noted the firmness or hardness increase with ripening and aging. This highlight formulation, fermentation condition, and maturation time all play critical roles in texture development. So, we discussed about the Brix value. From the graph, we can see that the Brix value for coconut milk increased from 12 to 13 after being converted into cheese. This suggests that the removal of water during the cheese making process result in higher concentration of soluble components. On the other hand, cow's milk started with a higher Brix value of 14, which further increased to 15 after conversion into cheese. This aligns with the known composition of cow's milk, which has a greater amount of lactose and proteins that contribute to its overall Brix value. Looking at the Duncan test result, we observe the sample with the same letter are not significantly different from each other. The cow's milk cheese MC had the highest Brix value of 15, followed by cow's milk with 14, and the coconut milk cheese show a value of 13, while coconut milk had the lowest at 12. This confirms that cow's milk has a higher concentration of sugar compared to coconut milk, as it contains lactose, which contributes to higher Brix value. Meanwhile, coconut milk, being lactose-free, contains glucose and sucrose in less amount. These results are consistent with previous studies, such as the one conducted by Costa et al. 2013, which state that the sugars found in a milk are composed of glucose and galactose. This finding highlights how different milk composition influence the final Brix value after cheese making. Okay, next, the slide presents the pH changes during the cheese making process. The graph shows a clear reduction in pH, which is expected due to fermentation. Microbial cultures convert sugars into lactic acid, lowering the pH and giving the cheese intangy taste while improving preservation. Coconut milk started with a pH of 6.07, which dropped to 5.173 after cheese making. Meanwhile, cow's milk had a slightly higher initial pH of 6.22 due to its lactose and protein content, which act as a buffer. However, after cheese formation, its pH significantly dropped to 4.353, the lowest among all samples. This drop is crucial because lactic acid bacteria must lower the pH rapidly to around 6.2 for proper coagulations and then further decrease it to around 4.5 to prevent microbial contamination. This ensures the cheese safety and shelf life. The Duncan test result confirm a significant difference between sample and particularly in cow's milk where the pH drop is more pronounced. This highlights the stronger acidification process in dairy cheese compared to coconut milk cheese. Okay, next goes to the fat percentage. This slide presents the changes in fat percentage during the cheese making process. Based on the graph, we can see a significant increase in fat content in coconut milk cheese from 38.47% to 60.97%, whilst cow's milk cheese shows a smaller rise from 6.49% to 8.49%. This increase in fat content is due to concentration effect, which occurs when a whey is removed during the cheese making. This process leads to a higher proportion of fat and protein in the final product, as explained by Lucy 2002, who states that weight loss during coagulation concentrate this component, resulting in a richer cheese. Additionally, research by Fernandez et al. 2015 highlights that the plant-based fat in coconut milk help accrete denser and more stable matrix, increasing fat retention in coconut milk cheese compared to dairy-based cheese. This is further supported by Sota et al. 2017, who emphasized the role of lactic acid bacteria in maintaining fat inside the protein matrix during fermentations. The Duncan test result confirmed a significant difference between the sample, with CMC having the highest fat content. These findings support the potential of coconut milk is producing high-fat plant-based cheese. The slide presents the changes in protein content during cheese making. Based on the graph, we can observe a decline in protein content for coconut milk cheese from 6.022 to 2.956, while cow's milk cheese show a slight increase from 5.838 to 6.062. One possible explanation for the potential loss in CMC is the isoelectric point of arginine in coconut milk, which is 10.8. Since cheese is made a lower of pH around 4.6, arginine does not coagulate well, leading to lower protein retention, cited by Halim et al. 2023. On the other hand, dairy-based cheese retains protein more effectively, likely due to the presence of casein, which is well suited for a coagulation at acidic pH level. This explains the slight increase in protein content observed in MC after cheese making. The Duncan test results indicate that CMC has significantly lower protein content compared to other samples, confirming the challenge of protein retention in plant-based cheese. This suggests a need for improved technique to enhance protein retention, making plant-based cheese more nutritionally compared to the traditional dairy cheese. Okay, now let's move on out to the result and discussion, specifically focusing on moisture analysis in cheese making. As we can see from the graph, the moisture content for both coconut milk and regular milk cheese decreased after the cheese making process. The initial moisture content of coconut milk cheese was 72.12%, which dropped to 66.77% after processing. Similarly, for regular milk cheese, the moisture content started at 89.07% and decreased to 80.52%. This reduction in moisture content is due to the separation of whey and curd, which is facilitated by lower pH environment. A more acidic environment leads to a better whey separation, which is turn reduce the moisture retained in the final cheese product. Additionally, based on previous research by Aini et al. 2019, the ideal moisture content for cheese analog is around 61.59%. The coconut milk cheese in our study falls within an acceptable range, but a higher moisture content could potentially shorten shelf life by encouraging microbial growth, as noted by Orjheba and Taiwo 2016. Finally, the Duncan test result shown in diagram 5 confirm that there is a significant difference between the moisture level of different samples. This reinforces our finding regarding moisture reduction during the cheese making process. This slide presents the analysis of texture changes in the cheese firmness. Based on the graph, coconut milk cheese exhibits the higher firmness at 220.43 G-force, while cow's milk cheese has a significant lower firmness of 33.9 GF. Before cheese making, both coconut milk and cow's milk had much lower firmness value, indicating a significant transformation during the process. The presence of medium-chain triglycerides, including lauric acid, plays a crucial role in the textural properties of coconut milk cheese. Real et al. 2016 identify MCTs in coconut milk and may attribute them to the dense texture of CMC. Additionally, MCT enhance curd firmness by forming strong connection with protein, reducing waste separation during filtrations. A study by Jaya et al. 2018 support this, stating that the high fat content in coconut milk helps create a superior structural matrix, making it a suitable alternative for cheese production. Moreover, Singh et al. 2021 further emphasized the role of MCT in improving curd stability and texture. These findings suggest that the coconut milk cheese may offer a firmer texture compared to traditional dairy cheese, making it a promising plant-based alternative. Overall, the results of this study support the use of coconut milk to create a stable, firm texture, high-fat cheese substitute. However, in order to enhance acidity and sensory qualities, further studies should be concentrated on optimizing sugar levels, protein structure, and microbial cultures. For the recommendations related with the food safety, further study need to be done related with the treatment for the lactic acid bacteria that use in the cheese for safety consuming. Furthermore, integrating consumer acceptability tests will further improve processing methods and offer insightful information about market potential, resulting in a more economical, viable plant-based cheese. Turbidity meters should use in further study to monitor the curdling process despite improve in understanding and control cheese making process.